So the lab technique I chose to do is hemaggluttination assay. This is a technique, it was invented by George Hurst in 1941-1942. He was an American virologist. And what it is, it's a method to detect viruses or bacteria and the red blood cells. It's simple, it's fast, it's easy, it can be used on large samples, so it makes it an ideal lab technique. What it is, it's a form of agglutination, which what agglutination is it's the clumping of cells in the presence of an antibody. So in the case of hemaggluttination assay, the viruses and bacteria are going to clump to the red blood cells. So the viruses and a lot of bacteria contain surface proteins and envelopes. And this is what actually sticks to the RBCs. And these viruses and bacteria, they bind to the N-acetylneuromanic acid. And like I said, that's what causes the binding. And when this is ran, a lattice structure forms from the binding of multiple viruses or bacteria to the red blood cells. And you can see this, which there's positive, negative, positive. That when you see the dark ones are positive, you can tell there's a presence of viruses or bacteria. If it's light like that, there's no presence. So for the procedure, the red blood cells can't settle out in suspension because the viruses or the bacteria are stuck to them. So in order to run this procedure, you take a dilution of the viral dilution or the bacterial dilution and add it to usually an equal part of red blood cell dilution. It could be less, it could be more, it just depends on what's being ran, and it depends on a lot of things such as pH and stuff like that. So usually it's an equal amount of viral or bacterial dilution to red blood cell dilution, and a good estimation of how many, well, not necessarily how many, but the presence of viruses or bacteria. It's cheap, it only takes 30 minutes, and it's an ideal lab technique, like I said. The lattice is what's counted. So, like I showed in the previous slide, if there's darkening, you can tell there's a presence of viruses or bacteria. But for bacteria, the technique is a little different. You would put it in an incubator for 30 minutes to an hour for optimal growth of the bacteria to see if there are bacteria present. So when the lattice structure is formed that's when you know when to stop. If you keep running it and kept adding more dilution, you would get less viruses or bacteria compared to red blood cell dilution. So if this happens, the agglutination won't take place. So sticking won't take place. You won't get an accurate reading of what's happening. So in conclusion, this form of viral assessment isn't as accurate as others, like plaque assay, which will measure the infectivity and how many there are and things of that sort. But all in all, it's a fast and simple technique, and it's good for the lab if you just need to see if there's a presence of viruses or bacteria. Anyway, this is my presentation. Thank you.